Hello, everyone, and welcome to today's Spud Smart Roundtable Series webinar. My name is Ashley Robinson, and I'm the editor of Spud Smart. Today, I'll be serving as your host for this webinar. And today's exciting theme is the future of Colorado potato beetle control, which I know you'll all be interested in, as we all have to battle with those, I'm going to say beautiful because they really are pretty, but horrible bugs. And today's presenters are Tim Waters, who is a professor, regional vegetable specialist, and Franklin County director for Washington State University Extension, and then Hannah Jean McCoy, who is a PhD candidate at the University of New Brunswick. And at this time, I'd also like to mention that this webinar has been approved to offer 1 CCA credit for those viewing live. We will show the QR code on the screen after the presentation, so you guys will have to make sure to stick around for that. And in today's webinar, you'll learn quite a few different things. Colorado potato beetles have wreaked havoc on potato fields for years. They feast on the leaves and leave little in their wake. The beetle is known for being able to breed insecticide-resistant offspring, making control of them an ever-evolving struggle. In this webinar, we'll look at some of the latest research work into how to control this pest and find out why wild potato relatives may just hold the key to future control. During this presentation, you'll likely have some questions for our presenters. So please type these into the chat box at any time during the webinar, and we'll address them during the question and answer session after their presentations. And today's webinar is being recorded and will be made available at spudsmart.com following this live event. Okay, so first up, we have Tim Waters, who, as I said before, is a professor, regional vegetable specialist, and Franklin County director for Washington State University. He has a Bachelor of Science degree in biology and a Master of Science and PhD degrees in etymology from Washington State University. He works with commercial vegetable producers in the Columbia Basin, where he focuses on integrated pest management issues on numerous crops. Tim conducts applied research in numerous disciplines, including entomology, weed science, plant pathology, soil and foliar fertility, cultivar evaluations, and irrigation management to address industry needs. And I'm going to bring Tim on here, and he's going to take it away with his presentation. So just make sure to unmute yourself, and please take it away there, Tim.
All right. Thanks, Ashley. Appreciate the introduction and excited to be able to present to the group today. I'm going to go through this presentation, and like Ashley said, if you guys have questions, you can put them in the chat box, and Hannah and I will answer questions, I think, at the end of both our presentations. So without further ado, I'll get rolling along here. Okay, sorry, there we go. Okay, so just a little bit about me and where I'm at. So this is a map of Washington State. I just think it's good to point out that I'm in a different area than many of you, I'm sure. So some of the things that I say will be regionally relevant to Washington. Other things will have a broader scope, so I'll try to point that out. I'm in the Columbia Basin of Washington, which is the area that's outlined there in orange. And the star is where I'm sitting right now. That's where my office is. That's where my research farm is. In the basin, there's about 1.2 million acres, 65 different crops that are grown in that irrigated region. And vegetable crops that I work with, they're worth about $1.6 billion annually. So with these higher value vegetable crops, pest management decisions are really important. And Colorado potato beetle is one of the important pests that we deal with here in the Columbia Basin. A little bit about Washington potatoes. We have about 250 growers, but we probably have fewer than that grow larger acreage. There's about 165,000 acres of potatoes in the state of Washington. Most of those are processed. We're known for growing high quality, high yielding potatoes, and we make up about 20% of the U.S. production. What we don't do is consume a lot of the potatoes in Washington because we have a really small population. So about 90% of them are sold outside of the state, and a good number of them are exported outside of the United States. Okay, so what I was asked to talk about here is a little bit about Colorado potato beetle biology. I'm going to talk about resistance and how it's developed over the years. And then I'm going to talk briefly about insecticide options with regard to Colorado potato beetle control. Okay, so the Colorado potato beetle, Leptinotarsa disseminata, it's the most widely recognized chewing pest of potato in the world, certainly. It's native to Mexico and was first found in the United States in 1859 and has readily moved into all parts of potato production, with the exception of not many found in Colorado, which is sort of interesting. Here in the picture, you can see a picture of the eggs egg cluster, which is always on the underside of the leaf. And then there's a first instar or neonate larvae on that lower part of the leaf where the lower orange arrow is. A little bit about biology. We're not going to go into this very deep. They do go through complete metamorphosis, which means that they go through a great transformation to go from the egg to the adult stage. So they have these larval stages, which do the majority of the feeding on the potato leaves, and then they pupate. And when they pupate, that great transformation occurs, and they go from that large squishy larvae to the adult that we all know and readily can identify. They're a big problem with potatoes because we have multiple generations per season. They overwinter as adults, and that overwintering or diapause is triggered by short days, cooler temperatures, and reduced food quality. So when those things start to occur in their environment, then the adults burrow into the soil and they diapause or go into a resting stage for anywhere from 3 to 24 months. They emerge in the spring, and that emergence coincides really well with the emergence of potato and nightshade plants that they feed upon. So what we'll see in this area is when we plant potatoes, when those potatoes start popping up, that's generally when the adults start to come out. One thing that's important to know about this emergence in the spring is that it's asynchronous, which means that they don't all come out at the same time. So that makes management a little bit more difficult as if, compared to if they all came out at the same time. So a little bit about insecticide resistance. So Colorado potato beetle feed on the leaves of solanaceous plants. Solanaceous plants have high concentrations of glycoalkaloids, which many species cannot digest because of the toxic nature of those compounds. And it's presumed that the evolution of Colorado potato beetle to detoxify these glycoalkaloids predisposed them with mutations in the population to easily detoxify pesticides. Being multivolting, which means they have multiple generations per year, and a high reproductive rate also predisposes them for the development of resistance. The more eggs that they have, the more likelihood that there's a genetic mutation in there that confers resistance. So with each female producing 600 plus eggs, there's a good chance of that. In 1952, a population of CPB developed resistance to DDT, and then dieldrin in 1958. So since we've been using commercial pesticides against Colorado potato beetle, they've been developing resistance. Almost all insecticides introduced in the eastern U.S. have populations of CPB that have developed resistance very rapidly. And this is just a chart that I borrowed from a website that Dr. Andre Aloykin maintains. And this chart shows the cumulative number of insecticides to which CPB has developed resistance and the years that has occurred. So you can see starting in the 1950s, we had one insecticide, two in the 1960s, and then it skyrocketed. And in the early 2000s, there were nearly 50 different pesticide compounds that CPB had developed resistance to. So this is a trend that's occurring and will continue to occur as we try to manage Colorado potato beetle with pesticides or insecticides specifically. So a little bit more on why CPB developed resistance to insecticides. So coevolution of the beetle and its host plants acquired development of the physiological capability to detoxify or tolerate poison. So like I said previously, they eat potato leaves or other solanaceous plant leaves that have glycoalkaloids, and they've developed the ability to detoxify those compounds, which predispose them with the ability to detoxify pesticides. A second is the high reproductive rate and the likelihood of having random mutations in the population that will confer resistance. So I'm not going to go into that in great detail, but the more eggs that they have, the more likely that they're going to have a mutant in there that is resistant to a pesticide. The third factor there is that beetles have a narrow host range. Both larvae and adults feed on the same plant, and this reduces the size of an unrestricted refuge where susceptible genotypes could escape exposure to chemicals. So although some susceptible beetles can survive on untreated volunteer potato plants within fields rotated or alternative crops or weeds, there's very limited numbers of those in some areas. In other areas, we have more abundant alternative hosts. So in those areas, we tend to see less resistance. So in the area that I am in, we have a lot of volunteer potatoes and we have a lot of solanaceous weeds, and we don't have resistance to insecticides, and that refugia is a big part of that. Fourth, with the exception of crop rotation, growers rely almost extensively on insecticides for beetle control. And so the more we apply these insecticides, the more that we aid that selection pressure for those genetic mutants that are resistant to those various insecticides. And finally, being a native to North America, the Colorado potato beetle didn't undergo a genetic bottleneck when it immigrated, or when they were, if they were to be brought into an area. So when they're brought into an area as an introduced pest, you have a bottleneck in fewer genes flowing around the population, so there's less likelihood to have resistance. But we don't have that here. So a little bit on insecticides, to which Colorado potato beetle has developed resistance or exposure due to exposure, in the laboratory or in the field. And you can see there's a lot of classes or chemical groups up here, and then it lists the common names of those pesticides to which Colorado potato beetle has developed resistance in the field or in the laboratory. So carbamates, which are still used to some degree in the United States, we still see that oxamil or methamil are used, and there's some resistance reported in the United States. Some old insecticides there that aren't labeled for use anymore. Again, organophosphates, there's a few organophosphates that are still labeled for use, many that aren't anymore, and resistant to those. Of course, the old organochlorines, which are not registered anywhere that I know, there's resistance to those. And kind of we can go down the line, but I don't know that it's necessary to read through all these, other than to maybe look at some of the newer ones, like we know, or relatively new in time compared to the organochlorines. But pyrethroids and neonicotinoids, of course, and then spinosins, and then even Bt. There's populations that have resistance to Bt. So we have widespread resistance to a lot of chemistries with Colorado potato beetle. So again, this points out that managing potato beetles with just chemistry is not the best and not a long-term sustainable approach. Okay, so a little bit on how to manage resistance or retain susceptibility. We want potato beetle to still be susceptible to pesticides. So the number one thing here is do not rely on insecticides alone. So relying on insecticides alone, like we've tended to do for the last 70 to 80 years, is why we've seen potato beetle populations develop such a high level of resistance to such a broad number of insecticides. So one thing we can do to not rely on insecticides alone is crop rotation. So that's really important. Where I sit in the Columbia Basin, generally potatoes are grown every 1 in 3 or 1 in 4 years. And so what that does is it means that those beetles that persist within that field don't get exposed to the same insecticide year after year. The beetles have to move to another field in order to find a host. So rotation is really important. So physical control is one option that helps. Potato beetles don't fly very much. They tend to walk from one host to the next. So physical control with things like trenches can be somewhat effective on small plots, but tend not to work in large fields because the beetles just tend to overcome that. Biological control can be an option to look for. Bavaria bassiana is one choice. And then if this is not a great picture of this adult beetle here, but you can see the little white flecks on the elytra or the wing covers, and those are tachinid flies that are a parasite of Colorado potato beetle. Another option is to alter planting timing. So we know that potato beetles come out of the ground pretty synchronously with nightshade plants. We can look at degree days, and if we have the flexibility, we could avoid planting during that major emergence period. So you could delay planting a little bit, and that would help somewhat with Colorado potato beetle. The problem with that is if you delay planting too much, in some areas you don't have time to get potatoes to maturity or to the size that you want. And planting early is not a great option because you put the plants at risk for fungal or bacterial rot as they sit in wet soil. And then another thing that Hannah's going to talk about is host plant resistance, which is something that's been worked on for some time and is still being worked on, and is probably going to be one of the options that we see going forward that is really going to help preserve susceptibility of the population. Rotating insecticides with different modes of action is extremely important. I'm going to talk about that a little bit here as we get into the slideshow, but this is one thing that we have been doing and we could probably do better. But I'll talk about that in more detail. The third point here is using economic thresholds for spray decisions. So, and really what I see, the most common mistake that I see in my area is people apply too early. And we really need to look at those thresholds and look at timing. And again, I'll talk about that a little bit more. Leaving refugia for susceptible beetles. So refugia could be weeds. It could be parts of a commercial field that you don't spray with that insecticide. And in the region that I'm in, volunteer potatoes are a refugia because volunteer potatoes occur in our wheat fields, our corn fields, our pea fields, and they usually don't get sprayed with insecticides. So that's a place where beetles can live and not be sprayed with insecticide for the first generation. That does cause problems as they move into the production fields, but it does also limit their exposure to insecticides for the first generation. And then the other thing that's really important that I focus on with my growers a lot and crop advisors is using the full labeled rate of insecticide. When we reduce rates, we reduce the likelihood of resistance developing. And I know that people like to save money by sometimes reducing insecticide rates. And when in terms of resistance management, that's not a very good practice. We want to stay near the full label rate or at the full label rate with most insecticides. And I want to note that I did take some of this information off of a website that Dr. Andre Loiken maintains. He's with the University of Maine. Okay, so one of the questions I get asked quite a bit is why is there not rampant resistance to insecticides with Colorado potato beetle in the western U.S. compared to the eastern United States? And this isn't a map of resistance. This is just an old map of where potatoes are generally grown in the United States. One of the things I would point out is we do rotate, and our growers are really good about rotating. We don't see potato on top of potato year after year very often. Our growers are pretty good about insecticide rotation. And then we have a lot of refugia. And I think that that's the one difference, particularly as our winters tend to not get as cold as they used to. We get volunteer potatoes pretty far across the production area. And those volunteer potatoes are a good place for potato beetles to persist in that first generation without being exposed to insecticide. So, and just a slide on judicious use of insecticides. Do we do that? And I think we mostly do that. And by judicious use, I say we don't want to use the same active ingredient more than twice per growing season. And with neonicotinoids, we tell our growers we only want to see one neonicotinoid application. The reason for that is neonicotinoids, particularly the plant applications, last for a long period of time. So when you make a second neonicotinoid application, it exposes multiple generations, and that's really risky in terms of resistance development. So when I say use the same ingredient not more than twice, we want to use those in sequence. And we want to use them back to back because, again, we don't want to challenge multiple generations to the same insecticide. So if we're going to use like abamectin or something like that, we only want to use it in the first generation or only in the second generation, not on both generations. We do not want to apply below label rates. I don't think that's normally a good idea. There are some instances where you might be able to get away with that, but it's generally not a good production practice. And then again, refugia areas. And like I said, we're fortunate that we have weeds and then volunteer potatoes, which people don't normally think that they're fortunate for that. Looks like I'm going to get going here a little quicker. Sorry. This is the insecticides that are labeled in the United States for Colorado potato beetle control. And I put some different colors on this slide to kind of highlight some of those that are the green ones are generally a little bit safer insecticide in terms of non-target organisms. The yellow ones are middle of the road, and the orange highlighted ones are those that are a little more toxic to non-target organisms. So you can see we have a lot of products that are labeled in the United States. So there's really no need to not rotate. I put lines through some of these. I took this from another author and then modified it a little bit based on my experience. So there's a few products there that they list that I don't think are very effective. And so I noted that with putting a line through the text on those. But again, in the United States and in much of the world, we have a lot of products that are available. And there's really not a good reason to not rotate insecticides. One of the things, and this comes from a paper out of out of Wisconsin, and it talks about rotation. Really important to rotate different modes of action. And they even break this down and look at this for multiple years, which I think is really important. So they've listed the year 1, 2, and 3 along the top, and then they've got in furrow compared to foliar programs or in furrow plus foliar programs. And they put together a nice recommendation of some different options you could do to control Colorado potato beetle. And think about resistance management, not just in one season, but for multiple seasons. And I think that if we're going to keep having beetles in our population that we control, this is something we're going to have to look at, particularly if you're not rotating from one field to the next. If you're putting potatoes in year after year in the same field, looking at multiple year rotations is really how we're going to keep how we're going to maintain the ability to control potato beetle with insecticides. And I think I'm running short on time, so I'm not going to go through these in great detail. But again, some good options here on some different programs that you could do over a 3-year period to keep a population of beetles out there that you can control. Okay, I'm going to go through these pretty quickly. I think I've got maybe 2 minutes left. I get asked sometimes about different application methods for neonicotinoids. And this is an experiment we did a number of years ago. And this is looking at the number of potato beetle larvae across the season. And the blue dotted line is the untreated checks. That's where we don't apply any neonicotinoids. The Admire Pro is the green line. That's an in-furrow application. The turquoise line is Cruiser Max insecticide applied as a seed treatment. And then the red line is Platinum applied at lay-by. And so you can see for the first generation, we get a big bump in the population in the untreated check. And then the second generation comes in and starts producing larvae that start to feed on the plant early July. And you can see that the untreated check line went up higher than the treated areas. But you can also see that those all kind of wore out of insecticide at the same time. And you can see the population develop in all the treated plots. So I just wanted to make this point here that, we know that applying these neonicotinoids are still effective in the region that I work, and the application method or neonicotinoid doesn't seem to make much of a difference in terms of control. So they're all still very effective in our region. And whether you apply them in furrow as a seed piece treatment or lay-by shortly after the potatoes have popped out of the ground, there's not much difference in residual control. Okay, so best timing for insecticides. Most insecticides for Colorado potato beetle control, you want to time at 50% egg hatch. And so the best way to determine that is by flipping over a lot of leaves and then looking at the color of the eggs. And color is not always a good indicator, but for potato beetle eggs, it is. They start out as very light yellow, and the closer they get to emergence, the darker color that they get. So obviously flipping over leaves, counting the number of egg masses, and then the proportion of those that have hatched is how you determine when to make that insecticide application. We want to time about 50% egg hatch because most of the insecticides are most effective on the early instar larvae, and the larger the larvae get, the more that they consume. So we want to control them when they're small. But if we apply at first egg hatch, then we have to make multiple applications to control the first generation. So waiting to 50% egg hatch ensures that we get a good amount of those eggs that are hatched, and that we minimize the number of applications we have to make per generation. The reason we can't do this all at the same time is because they emerge at different times and mate and lay eggs. So it's asynchronous emergence makes it a little bit difficult, but waiting for 50% egg hatch is usually the right thing to do. So again, just to cover my bases, do not apply anything more than 2 times per season. That's one of the ways that we see resistance develop rapidly. Do not apply the same insecticide to more than 1 generation per season, and do not apply at low or below label rates. And make sure when a product recommends an adjuvant, make sure you do that because it does make a difference with some of these insecticides for control. So out of control populations are super hard to control. Make sure you're on the ball. When multiple generations exist, nothing you do will get rid of the population with 1 application. So we get overlapping generations to some degree between first and second and third generation. And if you get an out of control population, 1 application is not going to control that. I'm not a big fan of tank mixes. They can only die once. You can make an argument in some cases for a tank mix, but most of the time that argument doesn't hold water with me. So summary continued. In the area that I work, neonics are still effective when applied appropriately. We don't want to use them more than once per season. We have a lot of products with good foliar efficacy. And one of the things we've seen our growers do with some success is perimeter applications. Potato beetles infest from the edge, and in large fields, oftentimes you can treat the perimeter with hanging the boom over the edge of the field or flying the edge of the field and get good control and reduce the amount of pesticide you have to apply on those fields by just treating those edges. And really scout your fields. There's no excuse to not scout for potato beetle. They're super easy to see. So I think I probably have used up my time in maybe a minute more. So I'm going to stop and then let Hannah take over.
Great job, Tim. Thank you so much for that presentation. And next, as said, we have Hannah up, but I'm just going to remind you guys, if you have any questions for our presenters, please ask those in the chat box. And when we're mentioning the chat box, we're talking about the comment section on YouTube there. So just type those questions in there. And then after Hannah's presentation, we'll bring both Hannah and Tim back on screen and we will ask them those questions. So next up, we have Hannah Jean McCoy, who is a PhD candidate at the University of New Brunswick. Hannah works in the laboratories of Helen Tai at Agriculture and Agri-Food Canada and Larry Calhoun at the University of New Brunswick Department of Chemistry. In 2022, she graduated from the University of New Brunswick with a Bachelor of Science degree with honors in medicinal chemistry. Since then, Hannah has been investigating Colorado potato beetle resistance in tuber-bearing potato wild relative species, Solane Okidae. So I'm going to bring Hannah on screen here and I'm going to let her present. So just 1 second, I'll get my slides off and add yours. Okay, there. Take it away, please, Hannah.
Thank you, Ashley, and thank you all for being here today. So as Ashley mentioned, I'm going to talk on my project that is centered around Colorado potato beetle resistance in a wild potato called Solanum okidae. So Tim touched on pretty much everything I'm going to with regards to the beetle, but the Colorado potato beetle are herbivores and they feed on solanum. So this would be potato, tomato, eggplant for their entire life cycle. However, plants have evolved to produce secondary metabolites, which are just a class of compounds and for defense against these herbivores. And what our lab is focused on is creating a hybrid potato with both commercial and wild relatives that have desirable properties to hopefully make one great super potato that has resistance to the Colorado potato beetle. So the wild species that our lab focuses on is Solanum okidae, and this is a wild tuber-bearing species that is native to both Argentina and Bolivia, and it has many desirable properties, including moderate blight resistance. However, the one that we focus on is its ability to deter Colorado potato beetle in the field. And I've also shown this in the laboratory, which I'll walk us through. So this was done doing a choice, no choice feeding assay and using second instar larvae. So I just have the different life cycles of the beetle down here, and second instar is just essentially the second larval stage. And the choice assay is essentially 3 treatments. So you can either have a no choice, which are shown on the 2 ends here, or a choice dish. So what that means is on the left side here, we have a no choice dish where the beetles are forced to feed on one species. So in this case, we have Shepherdie, which is a commercial cultivar, which the beetle really likes to feed on. And essentially there's 4 leaf discs overlapping, and we have the same over here for another no choice dish where it's Solanum okidae. So again, the wild relative that has shown the beetle resistance in the field. And in the middle here is where the magic happens, as I would like to say. And this is the choice dish. So we have overlapping leaf discs of both species. So on the diagonal here, we have Okidae, which is the O on my diagram up here. And on the other diagonal, we have Solanum tuberosum. So again, a species that the beetle doesn't like and the species that the beetle does like. So what I did is I took the second instar larvae and I placed one in each of the 3 treatment dishes and then left them to feed for 24 hours, which is what we see down here. So after that, the beetles were taken out of the Petri dishes, they were weighed, and we used a mobile app to record the percent consumed of each leaf disc. So it's a really handy tool that allowed us to get this data. And essentially what we have here is on the left-hand side, we have the Shepherdie no choice dish, which would be this one here. And as you can see, when not given the choice, the beetles were happy to feed on the Shepherdie plants. And in the middle here, we have the choice test. And as you can see again, when given the choice, the beetles consistently chose to feed on Shepherdie and not on Solanum okidae, which is what we also saw in the field. Most interestingly, though, when the beetles were not given the choice and they were forced to feed on Solanum okidae, the beetles actually chose to starve themselves as opposed to eat the plant. So that's what we have shown here. There was around 0% feeding on Solanum okidae, even though they weren't given a choice to feed on anything else. So that would be the equivalent of what is seen here. And the beetles coming off these dishes were generally smaller. They weren't producing as much frass, so not as much poop. They weren't moving around as much, and they generally just appeared ill, But most interestingly, they hadn't even taken a single bite out of the leaf disc. So this showed that Solanum okidae was capable of showing a very strong deterrence to the beetle, which was very promising for our lab. So with that, we wanted to figure out what was causing this resistance in Solanum okidae, and me being a chemistry student, I wanted to know what the class of compound was. So we did this by screening the foliar tissue of Solanum okidae for many class of compounds, and then hopefully finding a candidate compound from all that was produced in the leaf. And from there, we were able to identify cardiac glycosides, and these caught our eye because these compounds are known to limit heart muscle contractions, which makes them toxic to animals. For centuries, these have been isolated from the plant foxglove, which I have shown here, for the treatment of heart failure and cardiac arrhythmias. And two common cardiac glycosides that were originally isolated from this plant is called digitoxin and digoxin. And as you can see, these are larger molecules, which makes them very hard to synthesize. So to this day, these compounds are still isolated from plants as opposed to synthesized in labs for medical uses. And this was a really novel finding because previously it was not thought that cardiac glycosides were produced in potato, even in Solanum in general. So this was really promising for our lab. These compounds have also been noted as some component of some insecticides and rodenticides, and their general structure here is just a general steroidal core with a sugar molecule at this end and then a lactone ring at this end. And a lactone ring is essentially this little part of the molecule here that just contains the oxygen rings, and it's responsible for the biological activity seen in in humans. So with the target compounds in mind, we then wanted to look at isolating these compounds from our Solanum okidae. So just really simply to do this, we started with the plant, and it requires a lot of tissue in order to do this, but we take the leaf tissue, we grind it down, and then we boil it in this fancy apparatus here, and then we filter it and take the extract, and we're able to collect these colored compounds here into a jar, which we then can run on a fancy machine. But I won't walk us through what the machine does. I'll just say that we're able to compare the compounds that come off to standards like wabane and convolatoxin, and these are both commercially available cardiac glycosides, and I'll touch on wabane a little bit later, so just keep that in mind. But when we run our extract on this machine, we're able to isolate these peaks, and each peak on here is a different compound. So this is really useful because now we can take these compounds that are all separated out, collect them, and then we can do some cool science like figure out what the structure is, so we can confirm that we do have a cardiac glycoside, and then we can subject the beetle to feeding on these different compounds or forcing them to be in the same environment of these compounds and see how they react. So that's where I'm at with my project right now. However, we did do a different comparative feeding study using 3 different treatments. So a water control, Titan, which is a commercially available insecticide, and then wabane, which is a commercially available cardiac glycoside. So wabane isn't the compound we isolated from Solanum okidae. We don't have the complete structure yet, but our compound should be comparable to wabane in both size and function. So what we did here was we took a Kennebec leaf disc. It's a little different from what we had done previously for the feeding studies. So we took the leaf disc and then we dipped it in either of the 3 solutions. So either in water, Titan, or wabane on the bottom. And then we took 5-second instar larvae, so again, those prolific feeders, and left them to feed for 48 hours. So after 24 hours, the water controls started to have a lot of feeding on it as expected. The Titan had a little bit more than we would have liked from the point of beetle resistance. However, the wabane remained relatively untouched. And as you can see, these specks here are actually beetles that had already passed away. So they kicked the can, they were kind of out of the study. But this was promising. And after 48 hours, those differences became even more apparent, where the water control was essentially completely gone. The Titan had a lot of feeding, again, a lot more than we would like to see. And then the wabane, again, remained relatively untouched. And as you can see, the beetles in the other dishes are growing. They're getting bigger. They're producing a lot of frass. Whereas in the wabane, those that were still alive, at least, were small, They weren't moving around a whole lot. And again, they weren't even taking a bite out of the leaves. Apart from here, which isn't actually an outlier, this was more human error. And I didn't notice until, unfortunately, after the 24 hours that when I dipped this leaf specifically in the wobbing, that part of the leaf didn't get the compound on it. But this just goes to show further that the beetles will feed on anywhere that doesn't have the compound, but they completely avoid it once the cardiac glycoside is applied to the tissue. And this was similar to what we had seen when we forced the beetles to feed on Solanum ochidae. It was a similar behavior. And again, this was very promising for indicating that we did have a cardiac glycoside in Solanum ochidae. So it would be very unrealistic to take every plant we want to screen for cardiac glycosides and to collect the foliar tissue, extract it, purify it, and then do structured determination. I don't even want to admit how long it's taken me to get this point with one plant. So to do like an entire field just wouldn't be realistic. And because of that, I've developed a rapid method to screen for cardiac glycosides in potato. And this is done using a photometric assay called the Balget assay. And it's previously been used for detecting cardiac glycosides in drugs and foxglove, which again is that initial plant that it was isolated from. And essentially all it takes is a solution that undergoes a color change from yellow to orange when a cardiac glycoside is present. So all you need to do with this is take a handheld hole punch and take 4 hole punches per plant. So once you have your 4 hole punches, you just need to put those leaf discs in one well on a plate. So every well on this plate here is a different plant that we were able to screen. So once you put your leaf disc in the well, you add your solution of sodium picrate, which is a cheap chemical, readily available for purchase worldwide. And then you let it sit for 5 minutes, you remove your leaves, and then you can clearly see that there is a base level of color change for all the plants on this plate. So over here, we have just a blank solution of sodium picrate that didn't get any leaves added to it. So there's clearly a color change for pretty much every well, but there is a significant difference between some of the darker ones here. And those would be the plants that you want to select further on. So this is a really fast, easy, and inexpensive method that breeders can now use to screen for these cardiac glycosides in their potato and hopefully screen earlier on for beetle resistance than we currently do. So with that, the overall goal of my project is to create a commercial potato with Colorado potato beetle resistance, but more short-term, we're looking at creating DNA markers for cardiac glycosides in potato, and also looking at the biosynthetic pathway of cardiac glycosides in Solanum ochidae. So how are these compounds produced in potato, because this has yet to be discovered. And as I mentioned, finding cardiac glycosides in potato is a novel discovery, but also the target in the beetle has yet to be looked at for beetle resistance. So on both sides of it, it's a novel discovery. It produces a lot of opportunity for breeders and for entomologists when looking at Colorado potato beetle resistance. So overall, this project will hopefully decrease the amount of pesticides that have to be applied to our fields by creating a plant that is resistant to the beetle. Hopefully we won't have to apply as many pesticides, which is great in the eyes of the consumers, but in turn, this will also decrease the carbon footprint of breeders and provide an alternative to pesticides, which in turn will increase financial gain by saving on crop producing measures, as well as what was previously lost to beetle defoliation will increase our yields. And as I mentioned, cardiac glycosides are used in the medical industry from plants. So we still isolate these compounds from plants. So by creating a potato that is able to produce great tubers as well as have beetle resistance through cardiac glycosides, we now have a purpose for the vines that previously just went to waste. So this dual purpose crop provides a whole new market for potato and will allow us to create a new source for cardiac glycosides for the medical industry. So with that, I would like to thank everyone at Agriculture and Agri-Food Canada that have helped me, especially Helen, Pam, and Jess, who are the entomologists that helped me, as well as my advisory committee at UMB. And just an interesting little note, Dr. DeLongchamp's father was actually the first one to synthesize Wabane. So we've kind of come full circle here as he's sitting on my advisory committee. But with that, thank you, and I look forward to any of your questions.
Thanks so much, Hannah. You did such a good job. Really interesting work that you've been working on there. And so, guys, it's now time for the question and answer session. Just to let you know, we will be showing the code for the credits for the agronomist there on line right after we're done the question and answer session. But I am bringing back on Tim there, and we're going to get going with questions. So our first one comes from Kurt. And he was asking, how long of residual control do you get from I'm not going to try and pronounce that word in Washington. I'm thinking this is for Tim. Here in Manitoba, when spraying at early hatch, we can get one good season long control from one spray. And then he also commented after just saying that we are blessed with typically one generation per season. So
Sure.
I'll let you take it there, Tim.
Yeah, so I say it's cyan triniloprol. Those ones are really difficult to pronunciate, though. Yeah, so good question, Kurt. It depends on the application timing. So we've done some work with that product applied as a foliar application, and with a foliar application, we typically don't get much more than 2 weeks out of it. But again, it's going to depend on your environment a little bit. A lot of those insecticides are degraded by sunlight or photodegraded, and then also by microbial activity or metabolism within the plant. And the warmer it gets, the faster the plant's growing, the more sunlight, typically the shorter the residual with almost all insecticides. But if you've got one generation and you time it right, I could see how you could get by with that. And again, with most of our insecticides, we're timing that 50% egg hatch, and that tends to give you the best residual. So I know I didn't answer your question very directly there, but feel free to shoot me an email if you'd like, and I can send you some more information to get in a little more detail on that.
Thank you, Tim. And reminder to everyone, if you have any questions for our panelists, type those in the chat box, which is the comment section on YouTube, and we'll ask those. So next up, we have a question from Darren, and it's for Hannah. Hannah, have you or others in your lab looked at preferential feeding from currently available varieties?
Yeah, thank you for your question. Our lab is very small right now. It's mostly just me working on this. And no, not really. But this work is kind of from a previous entomologist who looked at wild relatives and feeding, Yvon Pelletier. So he has plenty of papers published. He's retired now, but yeah, I'm sure he's worked on it. But we mostly just focused on Sheppardy and Okiday for the time being.
Thank you, Hannah. And we have another question now from Darren for Tim this time. And he's asking, do you see diamide resistance in Washington? And have you worked with some of the new Group 30 products?
So no, we have not seen resistance to diamides in Washington. Like I said, we're in a really fortunate situation when it comes to resistance with Colorado potato beetle because of the way our production system works. But we have not seen resistance to diamides, and there are folks using diamides, and they do provide good control for Colorado potato beetle. In terms of the Group 30s, I have worked with them. I don't remember where I'm at in terms of agreements on what I can say about that, but the Group 30 class in general has looked good for Colorado potato beetle control. I don't think I can say more detail than that on those right now.
Okay, thank you so much for that information, Tim. And I guess we'll stay tuned to hear more about in the future your research on that. So we have another question from Rich this time, and it's beetles are attracted to stress plants. Should a well-balanced nutrition program early on in the plant's life cycle be considered as part of the beetle's BMP? Have labs considered that when testing? Hannah, I'll let you answer this first, and then Tim, you can come in with any additions.
I don't have much to say on that. Not my area of expertise. So maybe Tim, if you want to take it away.
Yeah. So, I agree. Stressed out plants tend to attract almost all the pest insects we work with tend to be attracted to stressed out plants. But I have seen fields that are, under really good conditions, good proper nutrition, good irrigation practices, still get attacked by Colorado potato beetle. So I don't think that it will not eliminate your problem, but the healthier the plants are out there, the more likely they are to have good lush, growth, and they'll be a little bit more resilient in terms of, that photosynthetic area that's removed, but it's not going to completely eliminate the problem. And I don't know about the laboratory testing and what they do there for that. But that'd be my comment on a field scale as to what I've observed there.
Thank you, Tim. And it looks like those are all the questions we have. So I am just going to bring up the code on the screen here. You guys can scan that. And if you are having any trouble, just email myself at arobinson@seedworldgroup.com with your CCA number in the email and your name. And I will make sure that you get that credit. And I'd just like to say a quick thank you to Hannah and Tim so much for presenting today. I really enjoyed listening to you guys. You guys were great and you did an amazing job. So thank you so much for presenting, and have a good rest of your day. And I will just do a few more housekeeping items. So if you have any troubles, just please reach out to myself and email me about that about getting that credit. And thank you to everyone for tuning in today. I really enjoyed learning about this with you. And again, a recording of this webinar will be made available on spudsmart.com. And thank you so much and have a great rest of your day.